I'm next going to jump into a little bit of a live demo. Now, I can't show it all. I'm going to keep it fairly high level, but I really want to push on two primary themes. The first one is benefits for scientists from daily usage of our Biologics LIMS. And the other one is the institutional benefits, the organizational value from having regular use of our Biologics LIMS. So, and I will try to pause at a few points to field questions, but I have to speed up just a little bit. Okay, so this is the dashboard for our Biologics LIMS. Landing here provides you a variety of insights about your existing samples, different types that you have in the system, their quantities, their states, as well as information about the freezers in which they are stored and managed. We also provide you insights into notebook activity. This is primarily oriented around your notebook activity, but of course, as I'll show you in a few minutes, when you actually go to the notebooks landing page, you can see all of the notebooks that are available in the system and do some nice searching for the information that's relevant to you. You can also examine your jobs list. You can know what work is needed, and between all of between the notebooks that you have ongoing in your jobs list and the various insights that are available, you can really understand what work is needed, how to prioritize that work, and so on. So as an analytical chemist or someone who's doing formulations development and stability studies or cell line development, and when I come to this page, I'm really reminded of what is ongoing and what I need to prioritize. So as a quick example, if I click into one of these jobs, I can find I can see all of the information about, in this case, this very simple set of steps that someone has asked me to run. I can click into this and see clarity around the sample information, the samples that are part of this. So I can see the samples that somebody has requested some information about. I can see its status, where those samples are. And I can also, in this case, the data already exists for one of these, so there's only one step left. But if I click into these, I can see the different types of assays that have been compiled by me in this case as I worked through this request for analysis of purified samples. Now, furthermore, as I think I kind of mentioned this in the Q&A, I can automatically connect this experiment data is automatically connected to the samples that I have listed here. So as I work through this job, as I worked through this request and imported this data for the set of input samples that were available, this information was automatically connected. For example, if I click on this processed sample 1, I can see a variety of sample details about where it was in the freezer, a variety of information about how, in this case, it was purified, as well as looking at its related information. So in this case, I can see information about its parent bioreactor harvest. I can see the expression system that produced it. I can see the molecule that we are targeting that's contained therein. And if I'm an analytical chemist, for example, it's going to be really important for me to be able to see a number of these physical property calculations about this related molecule. That way I can calculate concentrations based on absorbances and other such important analytical functions. I have similar abilities to explore the other facets of this. And you can, of course, see in this lineage graph how all of the different components of the registry and the sample management tooling are integrated. So as I showed you in that workflow screen, it is also true here that we have automatically connected existing assay data that was imported to this sample. In this case, I have a single run of SEC with summary information about my peaks that is maintained. I think we already talked quite a bit about assays in the Q&A of the last section, so I might pass through that a little bit swiftly. What I wanted to point out though about this particular job that I showed you and the way that we are aggregating these different assays is that this is precisely how we accelerate the aggregation of data for reporting. View this data and walk through its specifics. I can, as Adam talked about, kind of showed, I can make use of this data in my preferred analysis tools. And we're also, I'm really excited, in the middle of some really great cross-assay report development stuff. So I'd love to show that to people as it comes available as well. Let's see, there are other activities likely to be performed in the course of common work, such as registering new samples and other entities in the system. So, this is the sample type that I designed for my expression harvests. You can see a lot of useful information about this type of samples. And I might point out that as people move into some kind of systematic management of their data and samples, it becomes very valuable to automatically name a lot of these things and yet still provide common names. And we give you the ability to put in an automatic name and expression for anything that you register in the system. I can also see some insights about these samples, storage status, what are aliquots. Here I can see information about I can see information about these samples. What I want to point out, aside from the fact that I can aggregate information about the sample itself, the cell line, the molecule that it came from, freezer information, QC information, etc., is that I don't have to do any of the work when I register these in order to maintain that lineage, maintain that traceability. So as a quick example, if I add a sample here and I want to say that it came from a particular bioreactor and that it came from a particular expression system, let's say that I'm choosing expression system 1, and I'm choosing this particular bioreactor. When I create this, it automatically has all of that same information that I had before cooked up in here. So you get a very serious reduction of duplicate data entry and data tracking. I don't have to say which molecule this came from or which construct was used to produce this more than once in my life. We hear stories all the time of people going from massive multi-tab spreadsheets where you do have to maintain that information on every sheet, in every file for every sample of every type, and that of course takes up a lot of space and can be potentially error prone. Here, as long as you connect it to one thing in the system, it's connected to the rest. So this is actually a pretty decent way of giving you an introduction to our bioregistry. As you can see, there are nucleotide sequences, vectors, protein sequences, molecules, cell lines, all of these different, call them molecular entities that are critical things to track. And you can click through and explore all of these things. So as an example, if I come in and I look at, say, M1, as I showed you before, I can see lots of useful information about the physical properties, the structure format of these things. I can see all of the samples that contain this particular molecule. I can look at the protein sequences that compose this and their existing stoichiometry. For example, if I come in here, I can see a lot of detailed information about this particular protein sequence, as well as look at the sequence itself with all of the existing annotations, with all of the annotations that were either contained within the information registered in the system, or that you have manually added yourself after the fact. Let's see. So one thing that I want to hit on again, really hard here, is uniqueness in the system. So sequences in particular, but actually it's everything in the system. It's really important to maintain their uniqueness. So we have this sequence here, and even if you try to register this sequence with another name, we already know what it is. We will tell you so. That's true of molecules. If you try to register a molecule with the same stoichiometry as this particular protein sequence, then we will tell you it already exists. This is what it's called. This is all the data that's associated with it. And so we really emphasize that uniqueness. So then there is ELN. So as a most often daily, but at least weekly operation, you are not going to be able to dodge doing your documentation. But especially in the with respect to time, you are going to be able to dodge me dragging you through all, composing a full notebook from scratch. I think that instead, what I should do is show you a little bit more about our ELN from the perspective of amending a notebook and maybe creating one from a template. So here is a signed and locked notebook. This one is about toxicological characterization of some existing materials in the system. This is signed. This is something that you can export all of the data and the notebook itself for, in case it's something that you wanted to share, or in case you wanted to make use of the data outside of this system. This is locked, and it is easy to ensure that any way in which this notebook is used outside of the system, it's easy to ensure that it's either the same or not as the one that is stored in the database here. This notebook has a lot of information in it, references to existing samples in the system, attached files, information about methods, chromatograms. We allow you to add tables, files, images, new days to mark information in here, etc. All of this is through fairly expected text editing. One of the reasons why I want to show you amendments, though, is that we all, of course, make mistakes, either as authors or reviewers, but we've created a mechanism by which, even though it is critical that any notebook that is locked and signed stays that way, we've provided a mechanism by which you can make amendments to that, and we store, we maintain the versions of those notebooks. So in this case, you can look at this nice timeline where we say, hey, here's when things changed state. Here's when I made it and submitted it, when it was approved, when I amended it, when I amended it again. Now, I would hope that we would all only have to amend notebooks once, but we're all human. So its current form, its latest form, is of course this last one. But if I went back to, say, this state, this is the version that it was in originally, and you can see there's some differences. I included the wrong samples. one I made reference here. And if I come back to my current version, I can see the correct ones. Now, really quickly, just so I can show you quickly what this looks like. If I wanted to make yet another change, and I want to really quickly show you what it looks like to take advantage of the data connectedness of the system, I type / and I have access to all of the data in the system. In this case, I want to do something really simple like, make reference to, let's just add M1 and resubmit this for review. And I want to say, added molecule, and I can sign this. Oops. I have to choose someone to review this, and it's off. So hopefully that gives you a really quick version of what it looks like to amend these things. The other thing that I wanted to very quickly show is, if I want to create one of these from scratch, we have lots of existing templates. So you saw that tox characterization, it should look almost identical to the one that's in here. If this is something that you have to do all the time, you can very specifically shape what these notebooks ought to look like when you perform these actions, and generate a new one of these. Example, tox notebook, and create it. And when it's launched, it looks exactly like whatever the template said, including its references and everything else. So what we find that people very commonly do is essentially include instructions, like this little denotation here. Like, here's where you should add information about your sample IDs. Here's where you should add your chromatograms. Here's where you should add information about the reagents that you used, etc. Okay, let me briefly talk about some of the organizational value that you get from making regular use of our Biologics LIMS. We talked about the value of having a central source of information for all of your endeavors, and hopefully the few examples that I can jump to really quick here are going to help drive that home. So let's start with project planning and reporting. I hope you got the idea before, but from one of these sample grids, if I'm interested in asking various questions of my data, like what work has been done for a particular molecule, what methods have been used to purify that molecule, what samples do we have, and what are these samples, and what can they be used for, is really easy to do, both from the molecule itself as well as from these grids. So if I wanted to choose, see all the samples that are from a particular molecule, a particular cell line, that are in storage and are not aliquots, I'm going to immediately see all of that information. I have the ability to take action on these. I can request that something be done with these. I want you to purify these further, do some analysis for me. All of the, these can be added to pick lists. These can be added or removed from storage, added to jobs, all of those kinds of things. I further, for example, might want to, look at, the molecule that this came from, and I might want to ask a question like, what methods have been used to purify this molecule? Because someone asked me to generate some purified material from a particular harvest. Now, if all of my data were in lots of different systems, it might be difficult for me to, or at least time consuming, to figure out whether any of that work has already been done. But by simply clicking on this molecule in the same place where I can see all the information that I talked about before, I can see a set of notebooks that talk about it. Now, of course, I've built this to purpose so I can see, hey, here's all the purification method development for this particular molecule. And when I click into this, we take you to this notebook's home, and we automatically filter these the existing notebooks for anything that makes reference to this molecule. From here, since of course you might have more than 4, it might be 100. You can add further filters, time, include other references, authors, notebooks at different states, and use those to further filter down to just the notebooks that you actually care about, and you can extract important information about those particular methods. Another really important thing that I wanna briefly mention is projects. So I talked earlier in my slides presentation about the ways in which you can organize data. You've now seen this page about 6 times, so you may be familiar with it. I talked about how people working in one project, you might not want everyone to see all of the information in one particular project. So here I'm in my biologics multiverse. I'm in my biologics home area, but I have individual projects within this that I use to organize data. And in this case, as an example, you're going to watch this change. There are some specific project beta samples in here that are not going to be visible to people. They don't have specific permissions within here. It also filters out the noise generated from other projects, since information from this project is rolled up for a broader kind of project level view, should you want to see it. I'm really sorry, I'm kind of flying through this a little bit now. Let's see. I see one question here that's a bit of a sample management question. Can you reduce the volume of many samples at once? Yes, this is something that Hannah showed yesterday. When you check something out of storage, when you take it and make use of it, you're able to decrement those values altogether. One of us would love to show you more about that, probably after this demo. Let's see. Let me just conclude here. Let me just conclude here by let's see, one last message and conclusion, which is when you have all of this data integrated, when you're aligned on all of these conventions and the standardization of your data structures, it's really easy to see what experiments have already been performed, what data you have, what data you don't. It's really easy in, say, project sync meetings to decide what additional work actually needs to be done on one particular molecule or another. And it's easy then to aggregate data that's important to you for any kind of reporting or, say, IND submission. And by using a system like this at the organizational level, you have that shared language. But you're also really encouraging people to do lab work a little bit differently. I think more efficiently once you adapt to the enforced changes of working in a standardized system. And then I think ultimately that raises the data informatics intelligence of any organization that does this. I'm going to jump back over here and kind of conclude by saying that what I showed in the application today is exclusively some of its built-in features. Adam, of course, talked deeply about extensibility, and major extensibility is possible. I'm going to throw out one final invitation to come talk to us, learn more, let us learn from you, and I will. It looks like there aren't any current questions, but I'd really love to address any others that people have.